an audience question here from Sarika, who asks, with startups in mind, how do you balance the fine line between quick results and careful planning of assays to avoid hassles later? And Bill, you're on my screen if you want to take a crack at that one, or if
I think Sarika makes a very good point, which is it is a fine balancing act, and it's made worse by nobody gets as you get financing. The venture capital people tend not to write unlimited checks. There's a finite amount of funds available, and so it's not just the alacrity of getting the thing done, it's also the cost of getting it done. And having said that, there's not a really good answer to the question. I think the best that I can say is I think planning your approach to assay development as much as possible before actually stepping into the lab can pay off in terms of less time and money spent on false starts. But I think the other is so having a sense. This comes up sometimes with the earliest stage of tech transfer, which you sometimes don't even think of as tech transfer, where you're starting up with a product that came from Dr. Hotshot's laboratory at big name university, and the more you can extrapolate from Hotshot and his team, or her team, as the case may be, to get as much of a head start, I think is in your best interest. Actually, Cheryl's, I know, dealing with this far more directly than am I these days. But
Cheryl, you have any thoughts to share on that point?
Yeah, so I mean, I think that as a startup, you have to look at your potency, your identity, and your first, because that's going to be the major thing you need to develop. Most of our phase 1 or phase 2, we have developed the other safety tests, the contamination thing. So you really need to put that identity, because that's going to be unique for your product. So if you're going to think about what you're doing and developing something, that's where to develop it. And then as it transfers over, a lot of the other safety testing and suitability, your CMO or your lab that's going to produce your product should have a general idea on that. They kind of have set tests that they can bring in. They're going to throw, mycoplasma, all those kind of tests they're going to throw into their system that they have currently going. So really, it's the unique test for your product that you really need to focus on and have set to be able to do that tech transfer into the facility that's going to manufacture your first product.
Yeah. Thank you, Cheryl.
Actually, that is a very, that was a very good point, which I wish I'd thought of, which is particularly the safety testing. A lot of those assays actually are available as compendial assays, which means you can actually pull them straight out of the USP.
Oh, wow.
And then focus on the analytics that are unique to the product under development. Thank you, Cheryl. I wish I'd thought of that.
Write that down in the audience.
But those are only microbiological safety, right? When we talk about compendial, and even those are not the most advanced methods, right? But I think the safety testing, yes, those would have to be done first, because without those you wouldn't get any kind of agreement by the agencies to going into an IND execution. I think it's important to regard whether you do it internally or you're using some sort of a contract organization, that the documentation is really solid, right? That there is no requirement for any kind of method qualification. That's an industry practice, but it's not required. But even without a qualification, essentially, that we do in the industry, it needs to be solid. You need to have confidence in the actual data, and you have to provide some evidence of this method being suitable in your IND submission, right? And yeah,